Welcome to our unique hands-on edition where we dive into the world of bioinformatics tools. Today you'll learn practical skills using Clustal Omega, MegaX, Panther, David, InterPro KEGG, IGV, and PyMOL. Guide you step by step, showing how to align sequences, build phylogenetic trees, interpret gene enrichment, explore protein domains, visualize pathways, and examine genomic data in 3D whether you're a student, researcher, or enthusiast. This video is designed to make complex concepts accessible and engaging. Get ready to explore, analyze, and visualize biological data like never before. Let's jump right in and unlock the power of these essential bioinformatics tools together. Start with Clustal Omega, a powerful tool for multiple sequence alignment. Head over to bioinformatics.tools/msa/clustalo and paste in 3 or 4 FASTA sequences. With a simple click, Clustal Omega aligns your sequences, revealing similarities and differences. This alignment is crucial for understanding evolutionary relationships in functional regions. Once your alignment is complete, save the file for the next step. The process is intuitive, making it easy for beginners and experts alike. Sequence alignment forms the foundation for many downstream analyses, so mastering this skill is essential. Next, we'll see how to use this alignment in MegaX to build a phylogenetic tree. Now import your saved alignment into MegaX. This software lets you build phylogenetic trees, visualizing evolutionary relationships among your sequences. Simply load the alignment file, select the tree building method, and watch as MegaX generates a clear interactive tree. Phylogenetic trees help us trace lineage, identify common ancestors, and understand how genes evolve. The visual interface makes it easy to interpret results and customize your tree. This step bridges sequence alignment and evolutionary analysis, offering insights into genetic connections. With MegaX, you can explore the story behind your sequences, making bioinformatics both informative and visually engaging. Next, let's interpret gene set enrichment using Panther. Upload your gene list to PANTHER and run a geo-enrichment analysis. This tool identifies biological processes, molecular functions, and cellular components associated with your genes. The results highlight which pathways and functions are most relevant, helping you focus your research. Panther's user-friendly interface makes it easy to upload lists and view enriched categories. Understanding gene enrichment is key for linking genetic data to biological meaning. In the next scene, we'll compare these results with another popular tool, David, to gain deeper insights. I'll upload the same gene list to david.ncifcrf.gov. David offers a complementary approach to gene set enrichment, providing detailed biological process annotations. Compare the top processes returned by Panther and David. This comparison helps validate findings and uncover unique insights. David's interface allows you to explore enriched terms, visualize clusters, and download results. By using both tools, you ensure a comprehensive interpretation of your gene data. This dual analysis strengthens your research, revealing patterns and pathways that might be missed by a single tool. Next, we'll shift focus to protein domains and pathways using InterPro and KEGG. Let's explore protein domains with InterPro. Visit InterPro KEGG and paste your protein sequence. InterPro scans the sequence, identifying conserved domains and functional motifs. These domains reveal how proteins interact, their roles in cellular processes, and their evolutionary history. The results are displayed in a clear interactive format, making it easy to interpret domain architecture. Understanding protein domains is crucial for linking sequence data to function. In the next step, we'll connect these domains to broader metabolic and signaling pathways using KEGG, providing context for your findings. Switch to kegg.jp and search for the same gene. KEGG provides detailed pathway diagrams, showing how your gene fits into metabolic and signaling networks. Open the pathway diagram to see interactions, regulatory steps, and connections to other genes. Linking protein domains to pathways helps you understand biological context and functional relevance. KEGG's visual maps make complex networks easy to navigate and interpret. This step integrates molecular details with systems biology, offering a holistic view of gene function. Now let's move to genomic visualization with IGV and structural analysis with PyMOL. Download demo BAM or VCF files from NCBI and open Integrative Genomics Viewer, IGV. Load the human genome in your file, then zoom in on the reads. IGV lets you visualize genomic alignments, variant calls, and coverage in real time. This hands-on approach helps you spot mutations, structural variations, and sequence quality. IGV's interactive interface is perfect for exploring large datasets and making discoveries. Visualizing genomic data is essential for understanding genetic variation and its impact. Next, we'll bring your data to life in 3 dimensions using PyMOL. Finally, open PyMOL and drag in a PDB file. PyMOL lets you rotate, color, and explore protein structures in 3 dimensions. This visualization reveals residue interactions, structural motifs, and functional sites. Manipulating 3D models helps you understand how mutations affect structure and function. PyMOL's intuitive controls make it easy to highlight regions, animate movements, and export images. Combining genomic and structural visualization gives you a complete picture of biological data. With these tools, you're ready to tackle real-world bioinformatics challenges. Thanks for joining this hands-on masterclass. Keep exploring and innovating.